Welcome to part 1 of 3 looking at chapter 12, gene expression at the molecular level. So in this chapter we're going to be looking at gene expression, specifically how DNA is used to create different polypeptides. So it's gene function at the molecular level or at the cellular level. It's really important to remember that this relates to gene function at the level of traits, and that's because all the polypeptides or the proteins, those are the molecules that are going to give us our different traits, such as skin color or hair color, for example. So it's really important to kind of remember, we're looking at this so that you can understand how organisms are unique and how they get their different traits based on their genetic information. So this idea that our genetic material relates to our polypeptides or proteins, this idea was first proposed in 1908 by a guy named Archibald Garrod. And he was a physician working with patients. So he studied patients that specifically had different metabolic defects. And he noticed that these metabolic defects, they run within families. So he had this idea, proposed this relationship between the genes or that genetic material that's being passed down from parent to offspring to the production of enzymes that would cause these different metabolic defects. And he called these inborn errors of metabolism. So he thought that there's a mutation in a gene that would be inherited from one or both parents, and that inherited gene with the mutation would cause all these different defects that he was seeing in his patients. So Garrod, he proposed this idea, he had this hypothesis about genetic material relating to enzymes, but it wasn't until the 1940s where we have Beadle and Tatum, they did a really neat experiment using bread mold, so Neurosporsa crassa. And this is just bread mold that you find on bread if you leave it out in your kitchen or in your room. So this bread mold, it's really easy to grow because we can get it to grow just in our homes. And then they discover different strains of this bread mold, and they use those different strains in their study. And this study, again, you can read about it in your lecture book. I'm just going to quickly summarize it for the sake of time, but Beadle and Tatum, they concluded that a single gene controls the synthesis of a single enzyme. So they actually came up with evidence to support that hypothesis that was proposed in 1908. And they named it the one gene, one enzyme hypothesis. Remember, this is still a hypothesis because it just has a little bit of information to kind of back it up at this point. This one gene, one enzyme hypothesis since then has been modified because we have discovered new things. So one thing is that enzymes, like we learned in chapter 3, enzymes are only just one type of protein that can be synthesized or created. So because there's a bunch of different proteins, there's structural proteins, proteins used in defense, proteins used in transport, proteins used for movement, all those different proteins have to be coded for by genes as well, not just the enzymes. So genes can code for all these different proteins. The other thing is that some of these proteins that are created in cells are composed of one or more polypeptide or subunits. Specifically, again, if you remember from chapter 3, we talked about hemoglobin that we have in our red blood cells that transports oxygen. Hemoglobin is composed of 4 polypeptides. Those 4 polypeptides or subunits have to come together during that protein folding in order to get a functional hemoglobin protein. So based on this new information that we have discovered since the 1940s, we now have a one gene, one polypeptide theory. So here, instead of one gene, one enzyme, we have one gene codes for one polypeptide due to that information we just talked about. And then in addition, besides having a hypothesis, we now have a theory. And again, remember, theories have lots of support behind them, lots of experiments have been done, lots of evidence. So we're going to be looking at this one gene, one polypeptide theory. So to kind of summarize how we're going to take these genes and look and see how they actually give us our characteristics. So remember, our genetic material, it contains these genes, which are distinct units on our chromosomes and our DNA. Those genes are going to code for polypeptides. The polypeptide, one polypeptide can be a protein, or you can have multiple polypeptides come together to form your functional protein. And then the activities of these proteins, they determine the structure and function of our cells, because our cells, they all run on different types of proteins. And then those traits and characteristics of an organism are based on those cellular activities, so that structure and function of cells. So here we're kind of relating our genetic material, how it's used to make all these different proteins, and then those proteins give us all of our traits and characteristics. So kind of relating it to this bigger picture up here. So we're specifically going to look at how our genetic material codes for the polypeptides at that molecular level. So here we have a prokaryotic cell, and we know it's a prokaryotic cell because there's no nucleus, and we have a piece of DNA that is in a circular chromosome, and that's in the red. So we have a red piece of DNA in this prokaryotic cell. That DNA, it's going to be used to create an RNA molecule, and that's called transcription. So our DNA is going to be transcribed into an RNA molecule. And transcription, to transcribe something means to copy something. So we're copying our information from DNA into RNA. DNA and RNA, they're both types of nucleic acids. They do have a few differences that we learned about previously in this course, but they are the same type of language. So we're basically copying from one language our DNA into the RNA. Once we have that RNA, we're going to translate it. So this is the translation part. Our RNA is going to be translated into a polypeptide, and this occurs at the ribosome. So if you remember when we talked about cells, ribosomes are responsible for protein synthesis. So this is where our polypeptide is going to be put together in our cell. In eukaryotic cells, it's basically kind of the same process. You're going to have your DNA, so your genetic material, it's going to be transcribed into RNA. So again, DNA is used to create an RNA molecule. In eukaryotic cells, that RNA goes through an extra step called RNA processing, and we'll get into this a little bit more later. Once your RNA is processed, it can exit out of the nucleus into the cytosol, and that's where the translation part is going to occur. And again, that happens at the ribosome. We're going to use our RNA to create our polypeptide, or to put all of our amino acids together. Prokaryotic cells, everything happens in the cytosol because you don't have a nucleus in prokaryotic cells. In eukaryotic cells, transcription and RNA processing occurs in the nucleus, which makes sense because that's where the DNA is found within these eukaryotic cells. And then the translation part, the protein synthesis part, is done in the cytosol. So it's kind of a theme we've been seeing. Prokaryotic cells are a little simpler. Things happen kind of in the same area. Eukaryotic cells, we have that compartmentalization. So things occur in different parts of the cell. And we've seen this in photosynthesis and cellular respiration as well. So this transcription translation process that we're going to be going through, this is the central dogma of molecular biology. And that's because it really is the basis for understanding pretty much everything we research or look into in molecular or cellular biology. So this process is really, really important.